Hello everyone, I'm Ashley Heck, and I've spent around 10 years in neurobiology labs, largely exploring sex differences in the regulation of the neuroendocrine response to stress. And I'm absolutely thrilled to have had the opportunity to help test and develop the high-plex mouse neuropanel for CosMx here at Nanostring. It would have been so cool to have had access to this type of technology in my past neurobio research, and I wanted to share a quick potential application of this panel, which will be totally biased towards my own research interests. So I actually spent a lot of time in my PhD trying to better understand the regulation of a key network essential for hormone production and body-wide adaptation in response to stress. One player in that regulation is neurogenesis in the dentate gyrus of the hippocampus, which actually represents one of the very limited areas where this process continues to happen in adulthood. This is a highly dynamic process that is subject to stress, stress-related disease, and other changes in physiological status. And so being able to further examine the biology underlying this niche is something that would have been right up my research alley. With CosMx and this high-plex neuropanel, it's incredibly cool that we are able to examine interactions among cell types and transcriptional players within this highly localized neurogenic niche. We can see radial glial-like cells, which are the precursors for neuroblasts, which eventually become mature granule neurons. And I also have astrocytes highlighted here, and that's because there's a really nice body of evidence supporting the role of astrocytes in the control of the neurogenic process under stress. We can simultaneously visualize expression of FABP7 in white, which has been reported to be highly expressed in neurogenesis promoting astrocytes. And in purple, we have some of the many factors secreted by astrocytes that are known to influence neurogenesis. And so ultimately, by using CosMx, we're able to visualize transcriptional changes specifically in astrocytes that are likely to influence neurogenesis and potentially gain a better understanding of stress response regulation in the process. And all of that is done in a healthy young adult mouse. Now imagine how cool it would be to examine changes in these players during times of stress versus no stress, in males versus females, under pathological conditions versus healthy. Really, there are a lot of possibilities at play here, and I hope you find this neuropanel as exciting as I do.